All right. Hello, everyone, and thank you very much for joining us today. My name is Laura Newburn, and I'm the coordinator for the Center for Bee Ecology, Evolution, and Conservation at York University, or BC for short. This webinar series has been organized by members of BC, and after the presentation, we will have a Q&A period. So please ask your questions using our Q&A feature located at the bottom of your screen. If the discussion extends past the hour mark and you can no longer be present, don't worry. Both the presentation and the Q&A session are being recorded. And for speakers who give permission for us to share the recording, we will notify you when the talk is available and posted to our YouTube channel. So first, a little bit about BC. We are an organized research unit at York University. And if you'd like to learn more about us, you can visit our website, yorku.ca/bees. Or feel free to drop me a line. My email is bc@yorku.ca and I can direct you. We are an initiative that strives to advance research in the fields of bee ecology, evolution, and conservation. And the mission of BC is to foster interdisciplinary, innovative collaboration and cutting-edge research. This research is used for the advancement of knowledge and the implementation of policy changes to help sustain pollinators globally. Our overall goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and the vital ecosystem service they provide. So if you'd like to get our monthly newsletter, please subscribe to our listserv. You can send an email to listserv@yorku.ca with the following command in the body of the email: subscribe bees, and then your first name and your last name. By doing this, as I mentioned, you'll get our monthly newsletter, as well as any updates to events or research happening at BC. If you are a York University researcher or collaborator, this includes graduate students, please visit our website and apply for membership, or at least view our membership categories to see if you are eligible to apply. Below here, I have just a small selection of our BC associates, most of which are out doing happy bee research in the field. We have an exciting lineup of talks for our fall semester of the BEEc Speaker Series. This is a series that takes place one Friday each month, about the second or third Friday of each month at 11 a.m. Eastern. You can register for this series on our website, located here, BEEc/Speaker Series. We've already had one talk in September, which is posted to our YouTube channel. So if you weren't able to join us that month, please take out our YouTube channel and get caught up. And also visit our website to register for our November 8th and December 13th seminars as well. We'll have our winter semester lineup posted soon. So just a little bit of logistics about the BEEc Speaker Series today. We are using a Zoom webinar platform, so only panelists can turn their camera and microphones on. If you'd like to ask a question, please drop it into the Q&A box and it'll be posed to the speaker at the end of their talk. If you have any trouble, feel free to send a private message in the chat to me, the host, or email bc@yorku.ca. This presentation will be recorded, but it will have a delayed posting after the contents are published, and it'll be published on our YouTube channel when that time arrives. So here's the link to our YouTube channel, and you can see we have several videos from past talks, including BeeCon, our yearly conference, as well as previous seminar series, including our BST series and our internal associates talk. Before we get started today, I'd like to recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territories of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation, This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you'd like to know more about the history of the land that you come from, or where you're joining us from today, or where you conduct your research, this website, native-land.ca, is a fantastic resource for this exploration. It was initially a Canadian initiative, but now has worldwide coverage. And now I would like to welcome Dr. Gordon Fitch to introduce our guest speaker today, and he will also lead the Q&A session after.
All right, thank you, Laura, for that introduction, and thanks everyone for being here. I'm thrilled to get to announce today's BC Associate Speaker, Dr. Laura Figueroa. Laura is an amazing ecologist and entomologist who is incredibly passionate about biodiversity and sustainability. She says her overall aim of her research is to leverage science to gain a greater understanding of pressing environmental stressors and contribute to evidence-based solutions dealing with those stressors. She did her PhD at Cornell University, working with Scott McCart, and a National Science Foundation postdoctoral fellowship at the University of Massachusetts, where she is now an assistant professor in the Department of Environmental Conservation. She's worked in all sorts of amazing projects, including work on pollinator disease ecology, tropical ecology, climate change, and human-mediated invasive species spread. I think she's going to tell us about just a few of those today, but she's done also amazing work in around inclusion and equity in the sciences, and particularly promoting accessibility to graduate studies and encouraging people from historically marginalized groups to participate more in STEM. And currently, her lab is mostly working with bees and other pollinating arthropods. I think that's what she will be talking to us about today. So without further ado, Laura, please take it away.
Thank you so much, Gordon, for that lovely introduction. I really appreciate it. Okay, I'm going to share my screen. Can you please let me know if you are seeing my screen okay?
Yep, looks great.
Amazing. Wonderful. Well, it is an absolute joy to be here with you all. I'm so grateful for this invitation. And so as Gordon said, I am an assistant professor in the Environmental Conservation Department at UMass Amherst. And I would broadly describe myself as an ecologist, an environmental scientist, and an entomologist. And so while we're going to talk a lot about bees, I actually want to start broadly within the field of entomology. And often whenever I talk about entomology, there's a couple of different things that come to mind for different people. And so one of the first things, oh no, not slide advancing. Let's see. Okay, one of the first things that comes to mind are insects as disease vectors, which is absolutely understandable. When you think about Anopheles gambiae, the disease which vectors malaria, the parasite that causes malaria, 200 million cases worldwide, 1 million deaths a year. It can be a really, really big problem. Of course, another big area that comes to mind when people think about insects are agricultural pests that can be both damaging in the field as well as for stored products. And it doesn't just stop with agriculture. In Canada, in the Northeast, and many parts of the world, especially introduced insects can become pests of not only standing trees, but also stored timber products. And so often whenever there's conversations around insects, these are the three areas that get a lot of attention: disease and pests. But insects are also a big part of the solution, right? And so especially whenever we think about the broad evolutionary framework, we have a lot of insects that are predators. You have a lot of them which are parasitoids. You have a lot that can maintain healthy balance. And insects more broadly also are important for cycling nutrients in systems. And they're a super important source of food for invertebrates and vertebrates alike, including vertebrates such as humans, right? So throughout the world, insects account for a broad and multifaceted part of nutritional landscapes and provide many important nutrients as well. And when we think about food, that's a broad part of our culture. And insects just contribute and have contributed to millennia for our understanding and sense of place, for the materials that we use for art, for how we see each other and relate. And so insects are just really, really important. So I just wanted to, before getting into the world of bees, put a broader context for insects. But of course, a lot of us are here thinking about pollinators, and that's the area that I've been working on for over a decade. And the many ways that insect pollinators are important for not only agricultural crops, but also more broadly for wild plant diversity. When we're thinking about crops, though, one of the seminal papers that often has been cited thousands and thousands of times is Alexander Klein's critically important paper thinking about the important role of pollinators, where here on the x-axis I have what role or how important insects are, and on the y-axis is the number of crops. So you can see many have basically are not produced if there's no insects, many have high level, and so on. But one of the things that I want to start with is that while this paper was absolutely important and critical, there are, like any of our works, there are some limitations, right? And so it was really limited to crops that were present in the FAO, which excludes many regionally important species. Somehow, 2007 was nearly 20 years ago, in which there has been a lot of effort and advancements in the field of pollinator ecology. So there's been a vast amount of work. And this study does not evaluate quality metrics. It was really focused primarily on yield. And so within this context of introduction, I want to talk about a recent project that I have undertaken with some of my collaborators from Cornell University, where we wanted to actually fill in some of these gaps. And so we did a systematic review of the literature where we made a comprehensive search string of basically all of the crops present in the FAO, but also world crops, and screened over 28,000 title and abstracts, basically for finding any kind of, as many articles as we could find to really get a sense of what is the current role of insects for crop pollination around the world. And we ended up evaluating based on inclusion criteria, extracting data from over 1,000 studies. And we have a really broad, comprehensive assessment from all over the world. And basically what we wanted to do was see what is the impact for both quantity and quality. And so I'm showing you here some of, and this is preliminary, we're working on this now and finalizing, and we're hoping to submit this soon. So you're getting a sneak preview. And so what I'm showing you here at the top are the different metrics that we've evaluated in terms of quantity. And what you can see is that dashed line, if anything is touching that line, it means that we saw no significant effect. If it's on the right, it shows that pollinators improved that quality, that metric. If it's on the left, it means that it was negatively associated. And if it was touching the line, there was no effect. What you can overall see is that almost all of the elements are on the right. And so on the top, we have quantity metrics. So overall, having pollinators is really important for improving quantity metrics, but also if you look below, we have individual size and biomass level metrics, we have marketability metrics, we have nutritional metrics, plant growth metrics. For especially individual biomass and marketability, often pollinators are super important, though we have less evidence that they're consistently important for nutrition and plant growth, though there's some variability. But again, many, many elements in which they're important. We were evaluating these minor and major crops. So again, thinking major as in those that are present in the FAO, and minor as those that are regionally really important. And we can see that pollinators are really important at both scales. And for this study, we were really interested in evaluating the role of broad diversity of pollinators in these patterns. And so I'm presenting here basically the number of studies that we have that included these different types of orders colored by the class. And so basically we have green as insects, and we have birds and mammals, for example, in these other colors. But overall, if you just squint, it's mostly green. Insects are really, really dominating. And at the insect graph is we have to the species level, so not at the order. And what you can see is that almost all of them are bees. So a large proportion are Hymenoptera, though there's some important syrphids and Lepidoptera that are also present. Here we're showing some of the impact of specific groups. And again, bees are really, really important overall. Though, again, other groups, such as there's even some species of beetles that have been shown to have increased the benefit for pollination. And so all that is to say that pollinators are clearly very important in our assessment for the role of pollination, as everyone, most likely everyone in this call already thinks about. And so whenever we're thinking about why we have to monitor insects, there's some insects that are problematic, and we want to be able to understand and monitor to predict and control potential outbreaks. But then there's also many important groups that we want to monitor for evaluating potential evidence of decline and to evaluate the effectiveness of conservation measures. Because as we know, there's many groups that are declining and have been doing so for decades. And we really need to understand why, especially when we have lack of baseline data for a lot of these groups. And we need to know these factors in a changing landscape. So for many groups, including bees, the factors associated with their decline are interactive. So we can think about the interactions between pathogens, global change such as drought and heat waves, and overall average temperatures, pesticides, loss of habitat. And more than any one of these in isolation, it's the interactions. And so again, for the reasons that I've previously mentioned, we really need to have ways of rigorously monitoring and assessing the prevalence and abundance of these different groups. However, many of the current sampling approaches have limitations and known strengths. So for example, bowl and pan traps are very effective for getting groups of bees that you can identify and take to the lab and look for and identify to the species level, which for many species, it's very limited. You can't really easily identify them on the wing. So there are many times in which this is the appropriate level of sampling. However, there's a lot of times where this is not necessarily appropriate because we know that there are taxonomic biases. Not all groups are equally detected. Some are overrepresented, some are underrepresented from bowl traps. We know that they are lethal samples. So the reason for monitoring is monitoring species of conservation concern, potentially we would want to minimize lethal sampling as well as bycatch of other organisms. It requires a lot of people power to do this collecting, to clean all of the samples, to pin, etc. And broadly, if the emphasis, and it doesn't give us information about phenological patterns. It doesn't tell us when those bees fell within the 3-day period where the bowl traps were at. So there's known limitations. And we're currently living in a really interesting time, for better and for worse. We're living in the age of AI. And while, of course, there are many challenging and potentially deeply problematic elements, there are also potential strengths. And so some of you likely do have algorithms on your phone right now that use AI and can do so for conservation. So one of the most common approaches would be the Merlin app. So that's one where basically if there is, if you're hearing bird songs, you can take out this app and it can tell you basically what species of bird you're hearing based on the spectrogram that's produced based on its song. And this can be really important because it can be a non-lethal standardized way of getting information from these birds across different spatial scales. And also it has a science outreach component where people are engaging with the material in ways that are really interesting and important for conservation. And so within all of that, that's my way of introducing the concepts of bioacoustics. And now I want to get into some of the work that I've done in this field, as well as future directions that we're doing in my lab, which, as Gordon mentioned, is a brand new lab. So I want to start us off with a systematic review of automated bioacoustics models for monitoring insects broadly. Then I want to describe some of my efforts to develop a bioacoustics model for monitoring bees, specifically how we're using bioacoustics and other machine learning algorithms for monitoring bees in changing landscapes, with 2 case studies, some in solar facilities in the Northeast and some in tropical rainforests. And lastly, I want to introduce some of the projects that we have going on in the lab to invite opportunities for collaboration and engagement. So the reason I wanted to do this review is I wanted to see what has been done for evaluating like what models exist, right? Before we could move forward, we need to know what has already been done and build on that. And so with my technician, Anna Kohlberg, it was wonderful, and Chris Myers, we worked to develop a really comprehensive search string to find words that are associated with sound, with monitoring and sampling, all of the common names and scientific names of insects, and different types of machine learning algorithms. And so based on this inclusion criteria, we ended up extracting data from 176 studies. We grouped basically our types of models into 3 different categories. And so we started what we called SSO, which is basically any type of model where the researcher determines what is the variable of interest. So for example, if you have been studying crickets for a long time and you found that a certain frequency is associated with cricket species X, you're like, okay, when it's this frequency, we're going to make that association. And so the researcher is determining what the relationship is. But then you have the world of machine learning, where the researcher does not define the salient feature, right? So the part of why it's machine learning is that the model, the algorithms learn to determine those salient features themselves, and they're not predetermined by the researcher. And a subset of machine learning, which many of you may have heard, is deep learning. And so deep learning is a type of machine learning algorithm that uses neural networks, which is basically a type of processing for determining these features. And so we broke up the data into these 3 different groups. So basically, the researcher determines it's general machine learning or a specific type of deep learning, and we subsetted that out because it's really growing area. And so we wanted to see, is that where it's going into the future? And we did this for all insect groups. And so what did we find? It was really interesting that we found models for over 300 species of insects, and the most common ones were the red palm weevil, so a type of agricultural pests, honeybees in colony. So this is something that we're going to get back to. So it's within the hive, like the monitoring of activity within the hive. The third one was the rice beetle, another agricultural pest, and tied for 4th and 5th place was 2 species of disease vector. So really that broad representation of the kinds of reasons that insects influence human society, they were all represented. In terms of economically damaging pests, we found a lot of different ways that insect bioacoustics have been used to monitor them. So for example, it can be direct sounds that insects makes or indirect sound. So if you have, for example, a silo of grain, we found some interesting algorithms that were trained based on the movement of the grain. So ideally, if you have no pests and you have grain stored, there should be no sound. If you have a bunch of grain that is moving, you can train the algorithm to detect the presence of insects based on that indirect movement. And similarly, there are a lot of models for timber and forest products. For disease vectors, we only found mosquito species. There was no other group, even though there are other insect disease vectors. And really interesting work that some models could even detect up to 20 species of mosquitoes in the lab based on sound. So really great potential moving forward. As a conservation scientist, I was really excited at thinking about the ecological study and conservation applications. So a few of the broad areas that we found in our review was evidence for the assessment of restoration efforts. So basically whenever you implement certain strategy, is it better or worse? How do you tell? You have to have some way of monitoring. And so some of them, we found bioacoustics models that were used to monitor soil biodiversity efforts and restoration based on sound, tropical rainforest reforestation effort based on katydid calls, and even aquatic environments. So the aquatic soundscape and how the pond restoration could be influenced and monitored that way. It was really interesting that there were some that used community science efforts, and so the sampling that different people could bring, and based on that, detect the presence of an endangered species. And another area that was really fascinating with bioacoustics is there's a much broader history of bioacoustics used to monitor birds and bats. And once you have those recordings, there's no reason you can't use them for something else. And so we found an example of a survey of katydids based on a previous bat monitoring program. Broadly, we found that the vast majority of studies have focused on pests, some on ecologically relevant studies, and many fewer on parasites. And I have here in the lighter blue in the middle those honeybee hives, because again, there was a lot of research on honeybees, but almost all of them were focused on the hive. As far as the pests go, I just want to highlight that there was a lot of different substrates, and so just to have a broader perspective of all of the ways and attributes that this can be used for. Over time, we found that the number of studies in bioacoustics has expanded, and the proportion of machine learning and deep learning in particular has really, really expanded. And what that can look like can vary by order, where, for certain groups, certain types of algorithms were more used, but really there's a broad diversity, and it's really deep learning is the next, in many ways, it's the gold star is what we were finding. Because as we were evaluating accuracy measures, it had the highest measure of accuracy for classification, detection, and behavior. And so detection is basically presence absence. Classification is, okay, if there's these different sounds, this is a species katydid X, this is katydid Y, and this is a type of beetle, for example. That would be classifying. And behavior in our case was the presence of, for example, swarming in honeybee hives, that there's a specific type of behavior that's associated with a signal. With honeybee hives, also one element to note that was really interesting is that it could be used for monitoring other elements. And so there was a study that was finding that honeybee hives made specific sounds depending on pollutants in the environment. And so that the idea is that you could put these bioacoustic recorders in a honeybee hive, and that could be actually an air quality monitor, like a living air quality monitor, because the bees were making different sounds depending on the air pollutants. So that was really, really interesting. We found a few different gaps where the vast majority, so 83%, were focused on model development, and very few were applying it for conservation. So I highlighted kind of the ones that were actually applied, but the vast majority was like, this is a model that we could use hypothetically for doing these things. Very few made their code available for researchers, and even fewer had a user interface that could promote uptake without basically having a PhD in computer science. And as we have talked about, there's a lot of need. And so we really want to make sure that those who would want to use these resources would have access to. And the vast majority that were focused on bees were just honeybee hives, and very few were foraging on bees that were foraging, and very few were, for example, bumblebees and other species of bees. And so broadly, I just want to introduce that no way am I saying that bioacoustics should be the only strategy for monitoring. It just could be a part of a multifaceted toolkit for monitoring insects broadly. And a lot of us here really focus on bees. And so I wanted, within this context, to talk a little bit about my efforts over the last few years to develop a model to monitor bees. And so this is in collaboration with the Cornell lab of the K. Lisa Yang Center for Conservation Bioacoustics at the Cornell Lab of Ornithology. So Laurel Symes, Shyam Madhutsana, and Chris Myers, who is also at Cornell. And so the idea is that we can train these models based on the sounds that bees make when they're foraging, because the sounds can be converted into visual representation in these spectrograms. The spectrograms can be used to train convolutional neural networks, and these networks can allow us to detect and classify different groups of bees. So for example, queen bumblebees versus bumblebee workers versus honeybees. And we really have tried to be really intentional about collecting data across multiple years and different backgrounds, because I remember when I was chatting with Laurel early on, she shared a story of someone who was working with katydids and collected a lot of data, but all of it was inside. And you probably, wherever you are right now, if we were to be quiet for a second, you would realize, oh, there's the refrigerator's running, or there's some light, or there's some kind of background noise, and they accidentally trained the model trained on the refrigerator. So when they actually, after all this training, they went out to the field, the model didn't work because it was not trained on the katydid. It was trained on what was always present, which was the refrigerator. So we tried to be really intentional. We've been doing this across many years, many backgrounds, to avoid any kind of systematic bias. And these are kind of the broad types of classes that we've used: honeybees, bumblebees, and bumblebee buzz pollination, which, as you all have probably heard in the field, sounds very different. And I'll just show you a little bit of examples of how important it is to have mass amounts of data. So after the first year, here what I'm showing you is the precision and recall curves. So recall is basically of everything that's there, how many is your model catching? And your precision is how many of the ones that you're saying are a bee are a bee. So you can have, for example, be really precise. So everything that you say is a bee, and then you're missing a lot of things that you're not catching. Versus you catch every bee, but then you're also calling the plane a bee, the human voice a bee, etc. Oh, it's showing me that it's lagging. Can everyone hear me okay still? Can I get a thumbs up from?
Yep, we can still hear you.
Okay, great. I was just getting your internet is lagging. Okay, great. And so this is the precision recall curves for the first year of sampling. And then after the second year, we more than doubled our data set. And so the precision and recall curves are vastly improved. And we've continued to work on this. So this is after year 2, we actually have 2 more years of data, and we're finalizing the model now, so that hopefully we can even classify, like do the classification between honeybees and bumblebees, and more broadly, other groups longer term. And one of the elements that I'm really excited about is that we're going to integrate this into Raven Pro, so that anyone, including some of you who might be interested in monitoring using bioacoustics, you could just use this, basically put out the recorders, get your data, plug it in, and it would tell you when the bees were active in your given context. And so this is something that we're really excited about. And so just keep an eye out for this. Hopefully next year we can have this out for everyone. And so now I want to briefly touch on some of the applications of this work that we've been doing with bioacoustics and other types of machine learning algorithms. We'll start with some projects in solar. And so I assume it's similar in Canada, but certainly in the Northeast and in other parts of the US as well. There are big pushes to promote solar to meet renewable energy goals. And in a state such as Massachusetts, which is where I live and work now, it's a heavily forested state. And so there's really big questions about, like, what happens when you expand solar, and what's the relationship with the landscape context and the site characteristics, and whether people are planting pollinator habitat or not. So I really want to highlight my collaborators and the funding agency. This is a Department of Energy solar office grant. And so with my master's student, Grace Schifrin, we're really interested in understanding what the most common pollinators are in solar facilities in Massachusetts and New York, what the relationship is between landscape context and site characteristics, and whether we could use bioacoustics to monitor pollinators in solar facilities. So we selected 10 sites. We picked them out specifically to have a gradient of proportion of forest. And so this is what that looks like. These are our 10 sites in the proportion forest. We have, again, this nice gradient. And we sampled these sites using 3 different methods. So transect walks, quadrat sampling of the plants and the pollinators, and bioacoustics. And we've done this for 2 years. And we did this systematically throughout the season in different locations of the solar facility to have this broad representation. So from the first year of sampling, we found highly variable levels of abundance of pollinators, a lot of diversity, but the key players are really those honeybees, syrphid flies, and the common eastern bumblebee. So a lot of the broad groups that are dominant for the Northeast, which is unsurprising. For the preliminary assessment of year 1, we were finding this positive relationship between proportion developed land and honeybee abundance, which is potentially driven by a commercial and hobby beekeepers or others that are bringing in honeybee hives in for agriculture. But we weren't finding as many clear patterns for the other groups. But we have collected additional data this year, and we want to have a better understanding that hopefully will be a more complete story very soon. But whenever I talk about bioacoustics, people often ask, okay, bioacoustics is cool, but what about video? That's one of the, I get this question a lot. And so for this solar project, we also had an integration with video sampling to see what the similarities and differences are. And so this is with Yuki Hamada from the Argonne National Laboratories as a collaborator, where she set up this really high resolution camera system that can enable us to monitor insects at these facilities. And so you can get, like, this is some sample footage that we've gotten from the facilities. And so you can see clearly some groups of butterflies and other groups of insects that we found. And so we're really interested in evaluating and digging deeper into what the relationships are with the camera system, the bioacoustics, and the traditional visual observation methods. And so we're analyzing that, and we hope to have a more complete story for you soon. I also am interested in evaluating how we can use these technologies in different landscapes. So temperate forests, temperate regions in these modified solar facilities are very different than in other contexts of where I study. And thanks, Gordon, for bringing that I've done some work in the tropics. And so we're doing some work there as well, where they're also having these mass changes of landscape. It's just at a different scale and very different ecosystems. And in terms of bees, it's just a very different environment, right? So they're a lot more heavily dominated by social species, and particularly stingless bees that have really incredibly diverse life histories and foraging patterns, including a group of bees that I study, the one that forage on carrion. So there are groups of stingless bees that really primarily, there's 3 species in the neotropics that get their protein exclusively from carrion, as far as we know. And there's a growing list of species that we're finding that will get protein from both pollen and from meat, as well as the more traditional bees, as we think of these vegetarian wasps that only get their protein from pollen. And so in this context, I'll just, kind of giving some highlights of some of the work that we've done in this. We've looked at their microbiome and seeing what's happening with these bees that have such distinct dietary patterns. And we're finding that those facultative ones, not surprisingly, that are both going to pollen and to meat, have the most diverse microbiome, and both in terms of the number of organisms that are present in their gut, as well as like the compositional differences. And we most recently looked at functional metagenomics to see like what is going on, and we're finding certain types of characteristics associated with the consumption of meat. So all of that is broad, some of the work that we've already done, but forward-looking, we're really interested in sampling and getting a finer resolution of these foraging dynamics. And this is not bioacoustics, but camera-based systems where we can really monitor the interaction patterns of the bees on these different baits in a changing nutritional landscape. And we can even look at patterns of competition and dynamics, which we're really excited about. And so I want to highlight collaborators from actually, I'll go back to that slide. From La Una in Costa Rica, Quinn McFrederick from University of California, Riverside, and James Kroll from the University of Wisconsin-Madison, and the fantastic team, Costa Rica. And so kind of to close, I want to just talk a little bit about some of the future directions and projects we have going on in the lab to see, and I'm really focusing in on climate change and pollinator habitat, both separate and interact, how they can interact for promoting pollinator health. So we have a project in collaboration with Lynn Adler and Shalini Jha, and the postdoc on the grant is Kate Borchardt. We're found, Lynn and her colleagues had found previously that sunflower pollen and sunflower relatives are really medicinal for bees that are especially for common eastern bumblebees infected with a type of gut pathogen called Crithidia bombi. And so we want to know, okay, well, what happens in real life whenever people are planting a ton of pollinator habitat in Massachusetts and probably near Toronto as well. And so does having a higher proportion of Asteraceae plants in the planting actually influences these dynamics? And does that expand beyond just bombus impatiens, the dominant, the common eastern bumblebee that is a dominant in player? And so we're really evaluating that across 20 sites. And so Kate just wrapped up her field season about a month ago. And so she's in data analysis. And so we hope to have a more complete story about that soon. Master student in the lab, Natalie Beltran-Vargas, was really focused on the influx of like warming on plant pollinator interactions in the urban environment in particular. So her project was in community gardens in Holyoke, Massachusetts. new postdoc in the lab, Haley Schroeder, who just started, is really thinking about climate smart pollinator plantings in a different environment. So in cities as well, but not in the Northeast, but in the Front Range in Colorado. And so that's a project we're really excited about. And broadly, also thinking about, okay, what can we do about all of these changes of drought and heat? And so we're working with Diana Obregón and Katia Poveda at Cornell University, thinking about what are the relevant stressors that meliponiculturists, so people who work with stingless bees in Colombia, what can we do about it? Because we're working with Fundación ABC, who it's a nonprofit organization that really works with local communities, and they've been hearing from the meliponiculturists that drought has actually been a huge stressor for them and is really can like decimate their local operations. And this is not large commercial scale operations. Like, this is not multinational. These are smallholder people who often also have smallholder farms, and they produce this either for medicine for themselves and for small operations. And so really losing that could be a really negative impact for them. And so we're evaluating if there are strategies, like management strategies that we can implement during their drought to help them keep their colonies surviving over time. And then the last project that I want to touch on, because I know I wanted, I think I was asked to go until 11:45 to make sure that we had enough time for questions, is thinking about pollinator habitat within the context of climate change. And so this is a collaborative team funded by CASC, the National CASC Project, Climate Adaptation Science Center, thinking about different species that are often used for pollinator plantings that have broad areas of distribution and that may or may not be more adapted to heat waves depending on their region. And so we're really interested in establishing experimentally testing that, and to potentially make recommendations of should we evaluate and select genotypes from southern areas in the context of climate change. And so this is a very recently funded project. And so I am actually recruiting a postdoc for this to start next summer. So if you're interested or know someone I should talk to, please let me know, and I would be super happy to discuss this. And this would be, again, for starting next summer, and it would be a 2-year fellowship. And it's a program that's really interesting. It's with the National Task that also is associated with trainings. They do a lot of professional trainings. It's also associated with a national synthesis. And so I think it would be a really great opportunity if you know someone who you think this could be a fit for. And so broadly, I wanted to introduce some of the projects that we have, a lot of the methods and tools that we're developing. And so hopefully that can be useful for your research and your area of study in the world of bees. So with that, I'd love to thank my collaborators and my funding sources, and thank all of you for listening. And I would be super happy to take any questions.
Awesome, Laura. Thank you so much. That was a really fascinating talk. Hopefully, yeah, has people's minds going. So again, we'll use the Q&A function for questions. So please go ahead and post any questions that you have into the Q&A. I can moderate that. While we're waiting for questions to come in, maybe I can jump in and start. And I'm curious, when you were talking about, I know it's early stages, but talking about competition over those carrion resources for the stingless bees, what are you sort of starting to find there? Is this competition among species or between colonies within a species?
I think it can be all of them. And what's interesting is with carrion, it's an ephemeral resource. And so the fact that it can be really high in protein and very beneficial for them, but you're also competing not only with other bee species, but you're competing with flies. And one of the most wild things that I saw is some of the fly larvae, they grew up in mass and they develop. I didn't even think that this was possible. Like I truly, if I hadn't seen it, I wouldn't believe it. Within 24 hours, the eggs had grown into these like wriggling masses and like the actual meat substrate was lost. And so they're having to compete against that. They're having to compete against the bacteria that are being vectored and are also, secreting certain substances because like they don't want, like the bacteria like want to take, they want to take advantage of that. And so I think that there's a lot of different scales of competition. And so we're currently figuring out like the next steps for how to make that happen. And so I currently don't have a full answer, but what I can say is that it's a much broader framework of competition than what we usually think of just with bees.
Cool, thank you. Let's see, I'm not seeing any questions. Does anyone in the audience have a question for Laura?
I wanted to make sure I had enough space, but maybe I could have also gone more slowly. I'm happy to chat about anything, any of the projects, the bioacoustics, the research. Also, if you have any questions about being an early career researcher, I'm happy to chat about anything.
Hi, Laura. I'm here, Sandra Rehan. I can't start my video for whatever reason, but I really enjoyed your talk. I had a question about your work on the I think it was the project with Lynn and Chalonnet, the Asteraceae. So I guess, are you only looking at Asteraceae, or what would be the floral spectrum in that study?
Everything. So we're looking at these broad transects
Okay.
the season, and we are looking at floral abundance, blooming space, richness broadly, and then also diversity of Asteraceae, amount of Asteraceae. So because there's a potential that Asteraceae doesn't matter, like even though like it really matters in the lab, maybe just having a broader diversity of floral resources beyond Asteraceae is the better predictor of disease, or is the better predictor of the abundance and diversity. So we're evaluating beyond Asteraceae and just seeing does that or does not help explain the patterns.
Well, I guess we do similar kind of work in Toronto, and we find Asteraceae is just so predominant in terms of just, 3/4 of flowering plants that bees visit are Asteraceae. So, I guess would there be an experimental component to that? Like, are you going to because in nature you're just going to find Asteraceae? Is my prediction there.
Yeah, so in experimentally we found certainly that not only does that we found 7 species, we screened 7 different species of Asteraceae, and we found that 4 of the 7 had significant positive effects for the medicinal value. So we found certainly that experimentally that's present. We selected these sites to specifically include a range of proportion Asteraceae in bloom. And so we have these 20 sites picked to have a better gradient. And so while it's not experimental, we do have a gradient that we can evaluate across to hopefully see, at least at a level of correlation, whenever do you have a higher proportion of Asteraceae, do you or do you not see an impact on disease?
And last question, sorry, then are they naturally occurring variation, or was it because I know you guys have experimental stations there, would that be at the agriculture experiment station, or are that meadows?
The original project was we had this vision that we were going to be able to collaborate with people who were already establishing pollinator habitat. And we actually, we developed a salt pollinator seed mix that was species that were all native, perennial, and broad blooming, like would bloom throughout the season, native to the Northeast. And we have, I think, 4 or 5 of the 20 sites planted our seed mix. It just with the timing, so we that was originally going to be a treatment, and this seed mix is like very high in proportion Asteraceae, more so than a regular seed mix, which parentheses, most seed mixes that even say Northeast have a ton of non-native species. It just drives me wild. Like that's a whole problem, and I don't think we talk about that enough. But anyway, so we do have some that planted our seed mix. But a lot of it, really, I think in terms of the analysis, we're really going to evaluate it along this gradient. But we tried to plant ones that would be super high, and then there's wild, and some that are just mowing whatever's there.
Okay. Yeah, it's hard. I know.
It's hard.
Yeah. Long story short. All right. Thanks.
Great, thanks, Sandra. There's a couple of questions in the Q&A about the bioacoustic. So the first one from Shana O'Flaherty is asking, are there other fields of restoration ecology that you see this bioacoustic work being applied to? Shana's a
Totally.
restoration ecologist and curious to hear more.
Yeah, no, I think broadly it would be useful for many kinds of restoration ecology. I mean, we are planning on doing it in different kinds of meadows. I'm currently developing a project with Fish and Wildlife Service, and they're really interested in evaluating that in like their refuge system. I think that, and one of the things that I actually didn't talk about quite as much are like the limitations. So I think in urban areas, it can be problematic because when there's too much noise, like it can make the spectrogram hard to even visualize. And so the model wouldn't be able to be as easily trained. But I think in restoration efforts where like a lot of the noise are singing insects and birds and frogs and bats, the great thing about bioacoustics is you can look at all those simultaneously, right? So you could see in a restoration effort, like, are the bats responding differently than the birds and differently than the bees? And so I think in many different contexts of restoration, we could definitely, I mean, my hope would be that we would be able to use these methods.
Great. There's a follow-up question, which maybe your answer about the sort of broad range of applications of bioacoustic complicates the how you would answer this. But in terms of cost, how do you think of the cost effectiveness of something like bioacoustics versus hand trapping, which we know to be pretty cost effective?
Right. Well, it's interesting because like buying bowls and putting soap and water is super cheap, right? Of course. But the actual like people power of having to go and check them every 3 days and having to like pay people to like pin them and screen them and like there's a lot, and then like identify like to whatever broad resolution or weigh them if you're doing biomass. The ways of how we determine costs are actually broad, and often for a lot of projects, the most expensive part is the paying people. And I think if the idea is that you would be able to leave out these recorders for, let's say, 2 weeks at a time, or depending on the frequency of recording, a month at a time, and then you would be able to get this and like get the spreadsheet right away. That might be like, and you'd have to buy the recorder, which yes, each recorder is going to be $100, let's say, and you have to get the batteries and replace those every 3 weeks or whatever, and the memory card. But then like the memory card is like a one-time purchase, and so I don't think it's totally, I think the costs and benefits, it depends on what you're optimizing for. I think, again, if your question is species level resolution and like species richness of bees, the current model of bioacoustic models are not going to get you there. And I don't think we're ever going to get to species of all of the bees, but bioacoustics. Like if we can't even identify them on the wing, like I don't see a scenario where we would ever get to like Lasioglossum bioacoustic. No way. But I think we can get broader. And if like eventually I'm hoping that we could have like these broad classes that we include and then maybe have like an other. And so for a lot of models that I've seen, people end up having very coarse resolution anyway. And so I think when they're going to do coarse resolution, this would be more cost effective. I think similarly, there's questions around the video versus bioacoustics. And like the video gets you different levels of resolution, but it's much more data intensive. And so I don't think that there's a one size fits all, and it just depends on your question and your timeline and your availability of having people go to the field and process the samples.
Awesome, thanks. Another question is from A.K. Nixon about integrating bioacoustic data with data on other environmental variables. Have you looked at, like, can you improve predictions or use some combination of environmental data and bioacoustic data to better predict, say, population dynamics of pollinators in solar facilities?
Yeah, so I think the environment, we are looking at, we have some, it's not quite the same. So your question is about environmental variables. I think like the only thing like environmental variable example would be the one that I gave with the honeybees and like air pollution, and actually specifically linking a variable that can be variable in the landscape. But in solar, every time we went to the field, we made annotations about like the conditions of, proportion, like how much the weather and the conditions, and we have that tied in and connected with the bioacoustics recording. But I think maybe I'm not understanding the question. So it's like, how can we link it to these variables? I think we certainly can. I think because that's one of the strengths of bioacoustics is that we can look at patterns of like phenological patterns. And so you could see throughout the day, as for example, it's getting warmer, how is that being associated with bee activity, which is something that you can't get with bolt traps, right? And so I think that there are, or like if we know the time of when there was like rain or different elements of the environment, we could tie those much more closely depending on your settings. Because some people often do recordings of like every 1 minute or like 10 minutes of every hour, and so you could do it continuously. It just then takes more data. So it just, again, it's depending on what you're optimizing for.
Great, thank you. I guess one more question about, have you also comparing technologies? There's sort of another approach people are using is looking for eDNA, say on flowers, to detect interactions. Yeah, what are your thoughts about that as a alternative or complementary tool?
Totally. I mean, I think with eDNA, I think that there is a lot of potential for a lot of monitoring. I think that ultimately we need a diversified toolkit. It's not a one size fits all. I would love for eDNA protocols to be optimized to reduce the amount of plastic that is required to run those samples and those protocols. Just again, as someone who's like thinking through the lens of like waste and things like that. So I think that all of these tools need to be at the table, and depending on the question and the timeline and the approach and the funding, I think that they would all be really helpful.
I'm curious what you think about, you talked about plastics. I feel like there's also the carbon intensivity of machine learning as a technology.
Yeah.
Do you have
Totally.
feelings about
Yeah.
that?
So I think that I've been thinking a lot about this and how we sample. There are ones that are like in machine, like in device processing that require a lot of power. And like there's levels of like how frequently are you training and like how much do you need. So for example, like if we were using ChatGPT, like every time you run a search, it requires a ton of training and a lot of energy. With these, we have to optimize, and it takes a lot of data and a lot of power once we train the model. But once the model is trained, we're not rerunning the model every time you input your recording. Like you would have one, and then eventually, we could do regular updates as we have more data, but it's not that level of intensity for every single person who's using it. So it's not the equivalent of a ChatGPT. And so the idea is that could be really helpful for like minimizing those energy intensive needs. But it's complicated for sure.
Awesome. Thanks so much. I guess what looks like one last question. Are you accepting PhD students?
Please reach out to me. Reach out. Like the lab is growing and we have a wonderful, wonderful group. So please send me an email, LLF as in Frank 44 or as in Figueroa at umass.edu. And again, I'm recruiting a postdoc. So please come reach out for potential grad students, potential postdocs. I'd be happy to chat. Wonderful. Yay. Thank you so much,
Thank you
Gordon,
so
for the
much.
invitation. And thank you, Laura.
Yeah, thank you for the fascinating talk. And yeah, I think maybe we'll turn it back over to Laura wants to say some last words.
Absolutely. Thank you so much, Laura, for the very interesting talk. Especially learning about, what your lab is currently doing and what your future plans are as well. Very exciting stuff. I'd like to remind everyone that next month we do have another speaker, Dr. Matthew Betty, in our series of our BC Monthly Speaker Series. You can register for this event on our website, York University/Bees. And I'd also like to remind everyone that we have our yearly BeeCon event coming up next week. We have our keynote speaker, Dr. Chelsea Cook, an assistant professor at Marquette University, who will be giving our keynote talk on Friday. BeeCon is a 2-day event where we have a virtual session on Thursday, October 17th, and then we have in-person at York University or a virtual option as well on Friday, October 18th. Dr. Chelsea Cook will be discussing social animals and if they're able to manage a changing environment more effectively than non-social animals in the context of bees, as well as 41 additional talks centering around all things bees. So the registration deadline has passed. It was September 30th. However, if you do want to attend virtually, please send me an email to bc@yorku.ca, and I'll be able to slot you in for virtual attendance on either of the days. This is, of course, a free event, so we'd like to thank our funders, the Faculty of Environmental and Urban Change, as well as the Vice President of Research and Innovation at York University, who have been kind enough to sponsor this event and allow it to be free. And with that, I would like to thank you all for joining us today for another wonderful talk in our BC Speaker Series. And we look forward to seeing you again next month. Thank you, everyone.